So a couple of years ago, I went down a rabbit hole of trying to understand dementia a little bit more. It really started with trying to understand metabolism and cancer, and then metabolism led to dementia. And so here we are. This is really a very big overview of the deep dive that I did in this topic that I have to share. So this is, I put it out as a preprint, The Many Roads to Dementia, a systems view of Alzheimer's disease. So let's start with some historical background. We'll call it Tale of Two Dementias. As many of us know, in 1901, Alzheimer met a woman named Auguste Dieter at the Municipal Asylum for the Insane and Epileptics in Frankfurt. So she was 51 years old at the time of her admission, and she passed away in 1906 at the age of 55. And she exhibited very severe cognitive impairments, including progressive memory loss, disorientation and confusion, language difficulties, hallucinations, and paranoia. She was unfortunately a very, very sick individual. Alzheimer carefully documented her symptoms throughout her illness, and after she died in 1906, he preserved her brain tissue samples. They were analyzed in Emil Kreplin's lab in Munich. And one of the wonderful technological coincidences of the time was that there were some new staining methods that were developed just at the same time, 1 or 2 years beforehand. And they revealed several pathological features, like the amyloid plaques. So these are the extracellular deposits of a particular protein, amyloid beta. We're going to talk about it a lot coming up. He also observed neurofibrillary tangles, so NFTs. These are twisted intracellular fibers that are made of hyperphosphorylated tau protein. We will also talk a lot about tau proteins. He also observed cortical atrophy, neuronal loss. In 1907, he presented his findings at this conference, 37th Conference of Southwest German Psychiatrists in Tübingen, and also published a paper on a peculiar disease of the cerebral cortex. I'm not even going to try to attempt to read it in German. In 1907. Okay, so although dementia symptoms have been reported before, Alzheimer was the first to actually observe brain structural changes microscopically. And as I was saying, this was enabled by advancement in specifically silver staining techniques, which have just been developed. Let's say Bolshevsky silver stain. So it was developed just a few years beforehand, and it was crucial for visualizing all the amyloid plaques and the tangles. This is a figure from an excellent book that was really instrumental in shaping a lot of my thinking and understanding, Alzheimer's Turning Point by Jack de la Torre. So they were showing kind of the plaques. So it was called senile dementia at the time. The neural neurons, the NFTs, all of these wonderful things. It involves binding silver salts to abnormal protein aggregates, so they become visible under a microscope. And unlike some of the earlier methods, like Nissl staining, which only highlighted cell bodies, this method really revealed the fine structural pathology and abnormal protein accumulations. And that was qualitatively different compared to previous methods. And this is the technique that allowed the first clear visualization of Alzheimer's disease pathology. So this was really critical. In the late 1990s, that was kind of interesting that I found there as well, and early aughts. So her brain slides were actually re-examined with some modern immunohistochemical techniques. There was this 1997 study by Maurer and colleagues that confirmed, as Alzheimer has observed, the presence of amyloid plaques and NFTs in those samples. And then some later research suggested that she may have had this PSEN1 gene mutation, which is one of the penetrant genes that have been identified also in the late 90s as markers of early onset Alzheimer's disease. It seems to be an autosomal dominant early Alzheimer's disease gene. So this was kind of a sample from that paper. It's like, in summary, PSEN1 mutation in Auguste Dieter is consistent with the clinical findings and uncovers the cause of disease in this important historical case. So the the point of this particular analysis was not only to confirm with the modern techniques what Alzheimer has observed, but also to confirm that August Dieter actually had early onset Alzheimer, which, as we will cover in excruciating detail, is actually different from late onset Alzheimer's disease. Although there was this 2014 study by Rupp et al., and they were challenging this because they were saying, well, there was degradation of the tissue in the slides, methodological limitations. So they did not validate that she actually had the PSEN1 mutation. So right now we're actually not sure whether she did have that mutation, but everything else was consistent with her having early onset Alzheimer's. At the same time as Alois Alzheimer was doing his research, there was another person working, Oskar Fischer. And the same year, 1907, so he published his own study on senile dementia. That's what was the name that was used for what we call late onset Alzheimer's now. And his study was actually more comprehensive, at 16 cases. And he was focusing on elderly patients, so over 65, as opposed to August Dieter, who was obviously in her early 50s. But he also observed plaques in the aging brains and damaged neuronal processes surrounding the plaques. So he also linked plaque presence to cognitive decline, although he did not propose a clear cause at the time. He was at a less prestigious, maybe, university in Prague, which is now known as Charles University. So maybe that's one of the reasons why we really talk about Alois Alzheimer as opposed to Oskar Fischer in the same sentence. And so the origin of the name, who first named it Alzheimer's disease? Well, it was Alzheimer's lab director, Emil Kraepelin, and he was really a key figure in promoting Alzheimer's work. He's apparently a wonderful scientist. And in 1910, he coined the term Alzheimer's disease in his textbook, and who's really only talking about August Dieter's early onset case. Even though Oskar Fischer conducted thorough research with a larger sample, he did not get the same recognition. So maybe it was because he was in a less prestigious place, maybe because he was Jewish, do not know. But unfortunately, historically, his contributions are much less known. So originally, the term Alzheimer's disease was really referring only to the early onset cases, like August Dieter's. But over time, so Oskar Fischer also observed them, and this terminology really became popularized. So plaques were observed in both young and elderly patients with dementia, it seems. And it seems to have led to the merging of these two actually separate diseases, early and late onset cases, under a single name of Alzheimer's disease. And that caused also a lot of probably confusion in research in the subsequent years. Because early onset Alzheimer's, so it's largely genetic. It is linked to specific mutations that were identified, as I was saying, in the 1990s. So PSEN1, PSEN2, and APP genes. It's highly penetrant. That means that if you have these genes, you're very, very likely to develop this disease. But it does account for less than 5% of all cases. Unlike late onset Alzheimer's, makes up obviously the rest of the cases. It is influenced by genetic, environmental, lifestyle factors. It is actually modifiable. It is not caused by a specific mutation, although ApoE4 allele seems to increase the risk. I will also go into that in somewhat excruciating detail. And it is not fully penetrant. Like carriers may not necessarily develop the disease. Okay, ApoE. Now I'm just going to go into my ApoE story because I wanted to really dive into it. Although as a side note. So it's a protein, and it is involved in lipid metabolism and neural function. It is primarily synthesized in the liver, so it plays a very important role there in transporting lipids, including cholesterol and triglycerides. Within the brain, it is predominantly produced by astrocytes and to the lesser extent by microglia. Neurons generally do not produce it, but they do express receptors to take up lipids that are bound by ApoE4. So what do we have here? So kind of how I understand in general, what happens with ApoE? So yes, I said it's produced in the periphery and in the brain, carries things, so it binds to its receptors, like LDL receptor, so LDLR, LRP1, just receptors. At this particular moment, it's not important exactly what they are, but when it binds to them, forms an ApoE receptor complex. This complex gets internalized in the cells via endocytosis. And in there, so lipoproteins get broken down into cholesterol and phospholipids, and these are used in synapse function, myelin repair, neural membrane integrity preservation, a lot of very important, obviously, structural processes. After that, the protein itself, after it releases the cargo, like all of the lipids that it was carrying, so supposedly detaches and is then either recycled or degraded, and the receptor itself, so any of these guys, is recycled back to the membrane. That is what generally is supposed to be happening. Now, there are 3 generally recognized variants of this protein. ApoE3 is considered to be the baseline, and it is the most common one. So it has average risk of Alzheimer's, average affinity for the receptor, affinity, so how tightly it binds the receptor. Yes, average cargo quality. This will become important as I talk about ApoE2 versus 4. So the cargo is the lipids and the cholesterol. Like everything is, this is the baseline, causes average inflammation. Everything is kind of, this is a reference point. So ApoE2 actually seems to be associated with lower risk of Alzheimer's, as lower affinity for the receptor, so it's less tight binding when the protein binds to the receptor. Apparently has higher quality cargo, which means that there are fewer pre-oxidized lipids, which will obviously be in contrast with ApoE4. Slower cholesterol uptake, but there's no interference with normal function, so cholesterol is able to still function as it should in building cells, supporting synapse function, and all of those wonderful things. And it seems to be associated with low inflammation, so better for maybe low infection environments. Again, going to be important in a moment as we talk about ApoE4, which, on the contrary to that, has elevated risk of Alzheimer's. It has much tighter binding, so higher affinity for these receptors, more pre-oxidized lipids, so lower quality cargo, because, so the cargo is worse quality, but the binding is tighter. When the complex is internalized, the receptor is not recycled, but it is degraded. So we are not able to reduce this receptor in this particular case, or not as much. And in the long term, this can lead to decrease actually in receptor expression, because a lot of them are getting discarded instead of being reused. So again, and over time, that leads to lipid deprivation and decreased ability for self-repair, because you're not actually able to form these complexes that do have to serve a particular function. Insufficient cholesterol, then you have insufficient cholesterol for synapse function, insufficient myelin repair, cell stress, higher inflammation. But actually, higher inflammation is not necessarily a bad thing. And there was this really, really interesting study talking about how actually this is a gene variant that is expressed even now and used to be actually dominant in high infection environments. Higher inflammation is beneficial in a higher infection environments, or when you have a lot of parasites. But for long-term health, in a clean world where you do not have parasites living in your brain, that is not a very good thing. Okay. So I was writing all of this up in this preprint. This is where the figure is currently presented, and you can always go back and read through all of this. But I'm just trying to summarize everything I put into that paper. Okay, now we're going to go through the different hypotheses about the causes of late onset Alzheimer's. And as I said, I am really focusing just on this disease, not early onset, which is much more genetically driven. So let's start with the amyloid hypothesis, because that's something that everybody knows about, and that's something that both Fisher and Alzheimer observed in their early and late onset patients. Amyloid beta, it's a peptide, so it's a peptide fragment cleaved from the amyloid precursor protein, APP, which is a transmembrane protein involved in neural growth and repair. If you remember, APP gene is, mutation in APP gene is one of the penetrant mutations that actually has been associated with early onset Alzheimer's. Amyloid beta can exist in 3 general structural forms. It's monomeric, so these are single amyloid beta molecules. They're not typically harmful. Oligomeric, so these are the most neurotoxic. These are like small soluble aggregates of a couple of molecules, and also fibrillar. And these are larger insoluble aggregates that clump into extracellular plaques. So I found this figure here, kind of looking at some monomers, oligomers, and like what that actually kind of looks like. APP is so cleaved by 2 enzymes. Yep. This cleavage yields peptides of different lengths. So you have this going on. Some common forms would be amyloid, so beta 38, 40, 42, and 43. And the 42 one is the one that is most prone to aggregation and forms the core of these plaques. That's the reason I'm kind of going through this, because this is going to be important for trying to tease out the impact of different types of these things. By contrast, for example, the amyloid beta 40 is more soluble, less likely to form plaques. So this amyloid beta 42 one is actually been kind of biomarker of disease. And in healthy people, so the cerebrospinal fluid, CSF, fluid levels of it are about 500 picogram per ml. I just went down, like I was diving into this. Why just going into all of these details. And in Alzheimer's, these levels drop below actually about 400 picogram per ml, which is considered pathological. And of course, I was very curious. What do you mean it's associated with disease? Then why is it going down? Because I would think that if it is a marker of disease, maybe it should be higher. Well, apparently it becomes sequestered into plaques into brain tissue, and as a result, less of it is available to go into CSF. So if it is not trapped in the plaques in the brain tissue, more of it comes out in the CSF. If it is trapped in the plaques, then less of it has come out in the CSF, which kind of makes sense. And this paradoxical drop is now actually considered to be one of the key diagnostic markers used in clinical assessments of Alzheimer's. So this one I was just kind of looking at, trying to find the scoring. And this was, for example, so Tallet staging. It's a system for classifying the progression of particular amyloid beta deposition in the brain. And they're considered multiple phases. And one of the ways that they measure it and look at it is at the cerebrospinal fluid levels of it, which you can see are decreasing as the staging is increasing. Then I was like, okay, well, how do you measure them? And predictably, originally, they could really only be seen via postmortem silver staining. So you could only see it after somebody died. But in the 90s, there is this Pittsburgh Compound B methodology was developed and enabled in vivo imaging of amyloid beta in the brain, which, of course, makes it much more usable. It does not detect soluble oligomeric amyloid beta, which may be more toxic, but still. So this one, for example, this was an example that I was looking for. So Alzheimer's disease here, and healthy control is here, and then MCI, mild cognitive impairment. So non-converter, somebody who did not develop further on the amyloid beta, the Alzheimer's diseases versus the converter. And you can very clearly see, okay, so yes, you have a lot of these things here in those who actually then progress to disease. Also, in the early aughts, new assays allowed detection of it in the CSF and in the blood. And actually, one of the things now is looking at the ratios of amyloid, so 42 to 40. And this was actually the full figure that I was showing before. But they're not only looking at just decrease of the absolute, but also the change in ratios. Okay, interesting. So this methodology just binds to fibrillar amyloid beta, allows visualization through PET scans. Okay, so is it a good thing? Well, there's been a lot of evidence challenging the amyloid hypothesis, which does suggest that, oh, well, amyloid beta maybe is causing things. actually causing the deterioration. There are a couple of things here. So it seems like about actually a third of cognitively normal elderly show extensive amyloid plaques upon imaging or autopsy. Like some of the brains, you cannot distinguish if a person died cognitively normal or not, or healthy or not. So this was this was an interesting just examination of this. And from this paper, and they were talking that in this group of participants without clinically significant impairment, amyloid deposition was not associated with worse cognitive function, suggesting that an elderly person with significant amyloid burden can remain cognitively normal, which can suggest that maybe you do not need amyloid plaques to actually develop disease. And the opposite is also true. You have some patients with Alzheimer's disease who on postmortem autopsy do not have any amyloid plaques. So it's called plaque-negative Alzheimer's disease. So a lot of things here that are indicating that maybe it's not necessary, maybe it's not sufficient. Also, anti-amyloid drugs, at least so far, have been quite underwhelming. So this one, there was a big controversy with approving it a few years ago. Don't know if you were paying attention to that. But yes, it cleared a lot of the plaques, but it didn't seem to have very significant impact on cognitive decline. This one seems to be seems to do a little better, also reduced plaques quite significantly. It seems to have slowed cognitive decline but only, if you look 27%, oh, that looks great. But if you actually look at the numbers, some pretty modest delay, maybe about 4.5 months over 18 months. So I don't know, it seems still somewhat underwhelming. This one seems to be showing some benefit. Maybe will be showing benefit with long-term use. Results are still under evaluation. And some of the thinking is like, oh, maybe you are giving them too late, and maybe the disease is so advanced that you are actually not able to interfere with it. And that's possible. But it doesn't, I don't know, from the evidence that I see, it does not seem very whelming. So it seems like even in genetic early onset Alzheimer's disease, where you can start interventions before symptoms, they, at least so far, have not really prevented progression. So I'm in the amyloid skeptic camp, but I will explain that actually I feel the idea that it's more of a consequence of earlier damage than an initiating event. We're going to get to that. Also, some very interesting research coming out recently that amyloid beta may not be just a toxic agent. It actually might have physiological roles, which of course speaks to my evolutionary biology trained heart and seeing everything in the body has some kind of a physiological role, and in disease it's maybe out of balance or out of context. Obviously very common in cancer, which is my primary field, but maybe here as well. Like, it seems to be an antimicrobial peptide, binds to bacterial and viral surfaces, aggregates to trap the pathogens and maybe protect the blood-brain barrier. And that actually makes a lot of sense. So this is a paper, this Moore paper from a few years ago, where they're describing this may have a physiological role that then gets out of balance. So it might also act as kind of creating protective barrier at the site of damage. And if you're trying to plug leaks, for example, if something is happening with the blood-brain barrier, accumulating things, creating these clumps, not only can trap pathogens, but maybe even plug some leaks. And this suggests that also, like, one of the main side effects of these anti-amyloid drugs is brain bleeds, which might make sense if the primary function of these molecules is to protect the brain from bleeds. So if you're removing them, you're going to maybe get more of those. And of course, there is this Lesnaya scandal. Again, if you've been following the literature, you would see this. But there was this very influential 2006 study that claimed that there's a particular oligomer, so amyloid beta 56, as we all now know, that denotes the length of it. So it caused memory loss in mice, and when they blocked it, the mice could remember everything now. So it seemed like, of course, now we have a very wonderful thing. Well, 2022, the study was retracted to image manipulation. There was this very interesting report in Science really going through the fraud and how this was tracked down. So very unfortunately for a lot of this funding and a lot of these studies, it seems like this was really a rabbit hole that was leading in not quite the right direction. But there are some thoughts about amyloid. Maybe the issue is actually not production. Maybe it's the issue with clearance. Like the alternative theory is maybe it is produced normally, but clearance mechanisms fail with Alzheimer's. And there's some literature that suggests that yes, maybe this is something that may be happening. So in different cases. Also, role of sleep. We know that sleep deprivation can also lead to accumulation of amyloid beta in the interstitial fluids and fluctuations in CSF, like tied to sleep-wake cycle. So maybe disrupted sleep may impair clearance and contribute to pathology. I think there are a lot of interesting theories about the role and what might actually the more complex picture of what is happening with amyloid beta they're circulating around. But if clearance is the problem, maybe then removing the plaques does not fix the underlying issue, and maybe the disease will continue to progress because it's more of a systemic failure of brain maintenance systems. The systems that are supposed to be clearing this thing out are not working, so it's more, again, of a symptom of it. So then there is the tau hypothesis. And once again, trying to look at tau's normal function. So it's a microtubule associated protein found inside neurons. It has a normal role, has everything to bind and stabilize microtubules. So that's basically part of the neuron's internal skeleton. They're essential for maintaining neural shape and transporting nutrients, and generally have a lot of very important normal functions. Again, nature generally does not have any of this there by accident. And when functioning properly, they do seem to help ensure neuronal structure and efficient intracellular communication. Well, so some pathological changes that then happen is it seems like, at least as an observation, we're not talking cause or consequence, but just looking at the observation that in Alzheimer's, tau seems to become abnormally hyperphosphorylated. So that means that there are too many phosphate groups that are getting attached to the tau protein. And that causes it to actually detach from microtubules. So microtubules are no longer stable. So that interferes with intracellular transport. They also can misfold and aggregate, and so that's how we get these NFTs. And they, in turn, can accumulate in neurons, and maybe they actually now start to cause physical damage, like disrupt cellular function and synaptic loss, and eventually neuronal death. So this was a figure that I found in this paper, kind of talking about healthy versus disease, and the structural changes and stability changes that are then resulting from this from hyperphosphorylation and loss of function in tau proteins. Also, seems to be that these guys are not independent. That actually maybe amyloid beta may trigger or facilitate tau spread in Alzheimer's, because amyloid beta is often present earlier, and maybe it increases neuronal stresses and inflammation. So maybe that makes neurons more vulnerable to misfolding. Also, an interesting thing about tau, seems to act like a prion-like agent, so it has this self-propagating spread. So when it becomes misfolded, it can actually cause nearby tau to misfold, like prion diseases. And so that can actually now start, again, becomes a self-propagating issue. So it starts to spread across synaptically connected brain regions, and then acts as a template for more misfolding. So this is the mechanism that actually explains the spatial spread seen in Braak staging. So it's a measure of spatial progression in tau pathology. And some of the triggers include mitochondrial dysfunction, metabolic stress, chronic neuroinflammation. So maybe some other things are happening first, and then they are initiating this cascade. This is the thought to which I will come to again and again. Just kind of to finish up on this idea on the tau, because like in my field, for example, if you think that a particular mechanism of action or particular mechanism is causing disease, you're targeting that disease, and then disease is not modulated, that usually suggests that maybe this was not the right target, especially if you sufficiently engage the mechanism. And so just like with the clearing amyloid beta, it seems like, I mean, there are some tau targeting strategies, like there are some kinase inhibitors, there are some tau aggregation inhibitors that block the clumping, there's some immunotherapy. But from what I can tell, as of now, no really tau targeted treatment has really produced major results in clinical benefits and trials. So again, kind of coming back to this idea that maybe tau pathology is a downstream consequence and not the root cause of these problems. And the one that really makes sense to me, and this actually was my gateway, thinking about metabolism and mitochondrial dysfunction, that led me to deep dive into thinking about Alzheimer's as a metabolic disorder. And this is the competing hypothesis that actually makes a lot of sense to me, and I'm going to walk you through it, is that Alzheimer's is a fundamentally disorder of energy metabolism and vascular failure. And that is something that may actually be triggering a lot of these downstream effects. And this is also connected with a lot of the ideas that are put forth by Jack de la Torre in the book that I mentioned earlier. And this is a view that suggests that cognitive decline is driven by a mismatch between energy demands and energy supply, insufficient delivery and utilization of glucose and oxygen. And this kind of focuses on the idea of brain's energy need, mitochondrial health, neurovascular coupling. So not the protein aggregates at the root cause, but further upstream, and actually the energetic demands. So that is, if the neurons do not receive sufficient food, whether it's acute or chronic, we're going to talk about that as well, it can trigger this pathological cascade, so including NFTs, including amyloid beta accumulation, and that would actually then result in progressive loss of cognitive function. And the way that at least it's shaping up to, for my understanding of this, is that depending on where in the brain starts to be happening, then maybe you will have different deficits or maybe different types of dementia. The part of the brain that is going to be starved for nutrients is the one where you will start getting the deficits. Seems to kind of make sense, at least from my dilettante point of view. So kind of went to go in, so further I went into, okay, let's try to put some maybe numbers on this, or maybe try to get a better sense of this. And I mean, the brain is remarkable. It contains many, many billions of neurons and trillions of synapses. And this was a really, really exciting project. This work that came out, I think, a few months ago, when they actually took a slice of mouse brain tissue and mapped out all of the neurons and the connections. And this was insane. Like this particular cubic millimeter of the mouse brain contains more than 200,000 brain cells, 82,000 neurons, 500 million neuron connection points, 4 kilometers of neural wiring, all of this in a tiny block of tissue. And then, of course, the brain is built from a lot of these tiny blocks. Of course, as we all know, as we've all learned, despite comprising just 2% of body mass, it uses a lot of the total energy, primarily from glucose. Also, neurons are postmitotic, so generally they don't divide, although there are processes of neural regeneration, but they are more vulnerable to energy stresses. Another thing that happens is the cerebral blood flow decreases naturally with age. And that seems to be leading to also reduction in perfusion, especially, so according to this publication, between like 20% reduction between ages of 20 and 60. And of course, it becomes more dangerous when you are compounded by vascular disease and maybe insulin resistance and neuroinflammation. I'm going to get into all of these things. We all see these reports as well talking about high blood pressure being associated also with cognitive decline when it's chronic. So this really, to me, fits into this picture. And as a result of just reduced CBF flow, neurons don't get enough glucose or oxygen, especially if they're in high demand regions like the hippocampus, and that can lead to a lot of problems. And the way that we know that this is happening, again, we're not talking about cause effect right now as much, but we are talking about just the possible puzzle pieces that we will be assembling from here. But one of the ways to, of course, detect this is through PET imaging, positron emission tomography. So it scans using basically a fluorescent, like labeled glucose to track glucose uptake in the brain. And in the regions of the brain where the glucose uptake is high will light up because there's a lot of uptake here. Regions of hypometabolism will appear dimmer because there's less going on in there. And it would look like these giant machines. And of course, since neurons rely heavily on glucose, this will measure, it's a very good proxy for measuring synaptic activity and metabolic health. And as I said, regions with reduced uptake will indicate areas of hypometabolism, and that could lead to potential neuronal dysfunction. Do we actually see this? Well, yes, we do. So there's this very cool paper by Mosconi et al. From 2008. So what do we have here? So this is a reference. So these are patients with Alzheimer's. These are patients with mild cognitive impairment, so MCI, which did progress to Alzheimer's within 2 years. And these are people with MCI who remained stable, who did not progress. The images, so they show regions of reduced glucose metabolism. So that's what this is reporting here. The colors are normalized to actually show reduced metabolism, not elevated. Yeah, so it's saying color-coded reflects scores indicating severity of hypometabolism, not hyper. And so what do we see here? I mean, you don't even need an analogy. You can very clearly see it from the picture. People who, yes, they're having already some MCI, but who are stable, they have fewer regions where the glucose uptake is minimized, which is low. Those who progressed to Alzheimer's, now they have larger regions, and then people with full-blown Alzheimer's, you would just see there, yes, around the same places, but much more. And it does suggest, of course, that, yes, and it makes sense. If neurons cannot get enough food, you do not have enough glucose metabolism, that will start causing problems. Okay, so what causes metabolic dysfunction in the brain? Well, there are multiple things we can talk about, and I'm going to focus on one particular one, but we're going to talk about a lot of them in general. So mitochondrial dysfunction, that's one where basically you have impaired oxidative phosphorylation, and of course, and increased production of reactive oxygen species. And since the neurons do not use glycolysis, if the mitochondria don't work and they're not able to use oxygen well, they are in big trouble. Could be also reduced cerebral perfusion, like from vascular damage. So you would have less oxygen and glucose delivered to the brain. Again, the pipes are not there to get the food to the neurons. Of course, you could have impaired glucose transport and insulin signaling. So maybe glucose may be available, but maybe they can't use it efficiently. And that can be linked to some systemic metabolic disorders like diabetes, insulin resistance. Maybe some environmental things. So, for example, exogenous inhibitors like this one can compromise mitochondrial function. There was this very interesting experiment showing that essentially, the way that cyanide works, it's a cyanide analog, the way that it works is it blocks just one of the components of electron transport chain. So this was a very interesting experiment showing that this is kind of less potent version, essentially, of cyanide. So you're able to dilute it and gradually block metabolism, and you are seeing a lot of these effects here. Inflammation, immune signaling. So there could be caused by infections and chronic cell damage. And these are the things that I'm going to touch on as well. And of course, these are not mutually exclusive. They could, and often, I'm sure, do augment each other. The one that I want to talk about and really focus on, because that is really something that's been of great interest to me, is this glucose transport and insulin signaling and the connection between type 2 diabetes and brain function. Okay, so paradoxically, it seems like if you have high systemic glucose, why don't you have extra high glucose in the brain? Well, paradoxically, high systemic glucose actually might result in low availability of functional glucose in the brain. How would that work? So the brain is protected by blood-brain barrier. It's just a very thick, tightly regulated membrane, and it regulates chemical excess and ensures glucose availability. It's lined with GLUT1 transporters. They're insulin independent. And the idea here is so they have really high affinity for glucose, and they can prioritize brain needs even when systemic glucose is low. So insulin-dependent transporters, they require insulin to actually take up glucose. These do not. They want to make sure that whenever the brain needs glucose, it can have it. But if you have chronic hyperglycemia, so chronic elevated glucose, well, that can actually impair expression and function of these things. And that can lead to reduced glucose transport into the brain, even though you have high systemic sugars, high systemic glucose. And so as a result, brain glucose levels may drop or they become unstable. Apparently, it can also oscillate between excess and deprivation. And that can become a really, really big problem. Just kind of trying to find figures to illustrate these ideas. But essentially, so normal glycemia, you have your normal expressions of GLUT1, so glucose can go in whenever it needs to. But in hyperglycemia, the brain wants to be like, whoa, that we do not want too much, so maybe we'll downregulate some GLUT1s. Oh, it's oscillating now, upregulate, downregulate. This is not meant to be shutting up and down, and so eventually you might have a lot of glucose, and it will be compromising the functionality of these receptors, of these transporters. So this is one of the issues. Issue number one is chronic glucose oscillations can actually disrupt expression of glucose transporters in the blood-brain barrier. Okay, so now there's another thing that can happen. So these advanced glycation end products, AGEs, can lead to a leaky blood-brain barrier. So what are these things? Well, when proteins, lipids, or nucleic acids, when react with sugars, too many of them, they can form these structures. And that can be quite problematic because once they accumulate on and they can damage endothelial cells on the blood-brain barrier. So they can actually physically, it seems like, lead to structural disruption of tight junctions, increasing blood-brain barrier permeability. And this damage can also, of course, promote the release of inflammatory cytokines. And, well, if you remember me talking about, the point of amyloid beta is really to plug the leaks in the brain, or one of the things, trap pathogens, but maybe also plug the leaks. Now we're having these sugar protein clumps that are actually damaging the blood-brain barrier, starting to cause the leaks. Maybe that is not going to start triggering some of the amyloid beta formation and accumulation. And of course, leaks in there. This blood-brain barrier is not supposed to be leaky, so allows maybe toxins, some inflammatory molecules to enter, while also disrupting glucose transport regulation. So really a lot of bad things could be happening if the blood-brain barrier actually gets disrupted. So this was kind of a figure and illustration that I found, but essentially what I want to point out is, yes, we have these tight junctions, but they can actually start breaking down, and that can start causing a lot of problems. Okay, so the blood-brain barrier leakiness can cause glucose oscillations. We can have hypoglycemia, or we can have hyperglycemia, and that will cause maybe mitochondrial overload and stress. Neurons do not store glucose. They rely on oxidative phosphorylation. They are not meant to actually be subjected to all of these oscillations. When they're flooded with glucose, mitochondria can get overloaded. So you can get excessive electron transport chain activity, which can lead to reactive oxygen species. And then this, together with advanced glycation end products, can damage mitochondrial proteins. So now mitochondria don't work very well. And that can cause a lot of issues with ATP synthesis. Also, we, like mitochondria, used to be separate bacteria. So when they are tucked away in the cells, there's of course no immune response. But if they leak out, well, then maybe we're starting to see immune activation, because the immune system is now recognizing these bacterial components, mitochondria, which is now triggering a lot of maybe inflammation. And I was talking about amyloid beta and tau accumulation, which creates a self-reinforcing loop. We have metabolic stress, we have inflammation. So we end up with hyperglycemia, causing this, essentially inducing mitochondrial damage. So we have impaired ATP synthesis, we have release of these mitochondrial components that activate the immune system, and just really starting this vicious cycle of damage and inflammation. Coming back full circle to amyloid beta and tau. So now, what have we got? We have glucose oscillations. We have now leaky blood-brain barrier. We have advanced glycation end products, so that is starting to let things in. We're now having leaky mitochondria, and mitochondria are not functioning very well. You have oxidative stress, you have metabolic dysfunction. Well, that can now actually trigger some tau hyperphosphorylation. So now it detaches from microtubules and misfolds and propagates into toxic oligomers, and so it further disrupts axonal and mitochondrial transport. So now amyloid beta starts accumulating in mitochondria and further damages electron transport chain function, so increase ROS production, reactive oxygen species. A lot of these things, I hope you can start seeing how all of this can really become a giant cascade of amyloid beta and tau. They can impair the balance between mitochondrial fusion and fission, so it cannot repair itself very well. So now we have mitochondrial fragmentation, loss of function, again, disrupted transport, which leads to lack of proper ATP for synapses and for neurons, not for synapses, for neurons. Then starves them of ATP. So a lot of these things, they're really just accumulating and snowballing without one specific thing triggering it, but it's just picking up a lot of these damaged tissue along the way. Okay, I guess one more, astrocytes. So astrocytes are support staff. They support neurons, they convert glucose into lactate and then transfer it using astrocyte neuron lactate shuttle. Lactate is, so basically can be used this is the piece of the glucose that can serve as an energy substrate for neurons, especially during periods of high activity or stress. So it's like extra nutrient support. They can regulate neurotransmission by clearing excess glutamate from synaptic space. So usually they would prevent a thing called excited toxicity, which is when neurons are damaged or killed by excessive activation of glutamate receptors. But, well, chronic hyperglycemia can induce astrogliosis. So you have too many astrocytes activated and responding to stress. And that seems to limit their ability to efficiently deliver metabolic support to neurons, to regulate inflammation. So now you have a lot of the IL-6s and TNF-alphas and inflammatory markers. There's excess glutamate because it's not being cleared out. So that disrupts neurotransmission. That in turn causes a lot of other things like overstimulating receptors. You have excess calcium flux into neurons. So that is now going out of balance, which in turn can cause more mitochondrial stress, more reactive oxygen species, and eventually, obviously, neuronal death. So I hope you can see again and again like how a lot of these things are accumulating and feeding into each other. And I tried to put it together into a diagram and I probably missed some things. But like even with systemic hyperglycemia, maybe you're starting, as I was saying, formation of advanced glycation products, and then you have leaky blood-brain barrier, also with glucose circulation, and maybe now you have fluctuations in the brain, and maybe that starts to cause loss of small blood vessels, again, like you do have also in the rest of your body with vascular disease connected with diabetes. damage to astrocytes. So you can have some neuroinflammation that can cause more leaky blood-brain barrier. Then you can have more amyloid and that might trigger amyloid beta buildup, and that can lead to more mitochondrial stress, and mitochondrial stress can feed all of these things. So you have all of these complex feedback loops, which are just snowballing and accumulating. Whew. Okay, so that was talking about type 2 diabetes and glucose. There are other triggers of mitochondrial stress and neurodegeneration. It's not just all about blood sugar. Like, for example, traumatic brain injury just causes physical damage to vasculature. So this was an interesting paper from a few years ago about traumatic brain injury and risk of dementia in older veterans. And they were showing that TBI in veterans was associated with like really, really big increase in risk of developing dementia. Which again, kind of can make sense if the nutrient support delivery systems are compromised from whatever cause. It doesn't have to be blood sugar. It just keeps going and going. So you can damage vasculature. That can trigger, again, the calcium influx, lead to the ATP depletion, mitochondrial swelling, neural death. And that does seem to be associated with accelerated tau pathology. Again, or as we're all hearing about with the last few years, the brain damage in athletes. Strokes and ischemia, again, can cause acute energy crisis due to lack of oxygen and glucose. There's such a thing as reperfusion injury, which can flood the damaged tissue with oxygen. So, maybe now you start having excessive reactive oxygen species generation, which can, in some cases, maybe lead to more mitochondrial membrane damage and inflammation, and start triggering all of some of these pieces, which if they catch on can start accumulating. Infections and neuroinflammation. So that was pretty interesting. This one I think we've seen the HSV1, so viral reactivation has been linked to amyloid and tau accumulation, especially in APOE4 carriers, which are more prone to higher inflammation. That used to be a defensive mechanism, but now, of course, it is now another trigger. Disease like gingivitis has been found in the brains of people with Alzheimer's disease, and it also may promote amyloid accumulation. But in general, neuroinflammation. If you think back to that hypothesis, to that diagram, you pluck on that string, and whatever other cascade will start going from there. Lyme disease, sepsis, pneumonia. Like a lot of the chronic diseases, things that are just the chronic inflammation, they can trigger that and again start that process. Long COVID seems to be associated with persistent blood-brain barrier disruption, inflammatory brain injury, and really just generally any kind of chronic inflammation, if that gets into the brain, it doesn't necessarily, but it can trigger these processes. Some of the mechanisms that seem to be kind of popping up in the literature are systemic infections, which can lead to cytokine release, which can maybe cross the blood-brain barrier, which can then cause some mitochondrial impairment and plug into this whole neural energy crisis theory. And another thing that could be happening, don't know if that actually happens, but at least mechanistically it could, because immune cells, they have a lot of energy demands. So maybe they divert glucose, reduce availability of that for neurons, basically taking up these resources, so might compound energy failure. So I was trying to think through, basically that's why I was calling this the many roads to dementia, that there are multiple things, like there are a lot of things that could be happening in that, with that figure that I showed earlier, but then other things can trigger it, not necessarily the glucose, but maybe infections will trigger the neuroinflammation. Maybe a stroke will then, or injury, or some kind of reperfusion injury, interfere with cerebral blood flow. Polypharmacy has been actually shown to, I don't know if it would be the same mechanisms, but also something's been in the literature. Maybe something is affecting mitochondrial function, why polypharmacy can lead to symptoms that may look like dementia. Luckily, very reversible. And then now finally, to the last piece of this never-ending story, the treatment and prevention strategies. The key domains that I've seen in the literature, of course, metabolic regulation, systemic and oral infections, sensory health and medication management. There are some experimental strategies that I'm going to cover, which seem to be very interesting. And also a little bit about early detection and digital tools and like AI involvement in predicting who might be on the path to developing issues. So let's start with kind of the basic ones, the anti-diabetic medications, my beloved SGLT2s, which again, I'm not involved in with any company who does this. I have no financial interest here, but I'm just amazed at how cool this mechanism of action is. Which is, so they block glucose reabsorption in the kidneys, and essentially you pee out the glucose as opposed to have it go through your system first and cause all the damage. So there was a study that I saw last year, population-based cohort study, and they found that 35% lower risk of dementia in type 2 diabetes patients who were actually managing their glucose compared to this other medication. I did not see it relative to controls, but I mean, that is pretty remarkable. Some studies are looking at GLP-1s, those semaglutides and tirzepatides and all of those things. So I do not know if this is, so some of, like this was a study from a few years ago, whether talking about how it is associated maybe with lower incidence of dementia, whether it's indirectly from normalizing glucose and insulin versus some additional direct effects. It remains to be evaluated. I've seen people talk about how actually there are direct effects, but those people have developed, work for Novo Nordisk and have developed semaglutide, so a little bit of a grain of salt there. Did not see like definitive experiments talking about that. Maybe something to be designed, but even if not, basically anything that if you normalize glucose and solve and normalize metabolic problems in itself, that is already quite remarkable. Infections. I mean, we talked about systemic infections. They seem to be associated with increased risk of long-term cognitive decline. But if we're looking at my big diagram of everything being interconnected and triggering everything, maybe this is really something that triggers just basic a systemic inflammatory response. And if that does cross into the blood, does cross the blood-brain barrier, which may be leaky for some other reasons, then might induce neuroinflammation. And as I was saying, maybe activated immune cells, they shift to glycolysis during immune responses. They need energy fast, even though it's not very efficient. So they might be diverting some resources from neurons, leading to neural energy crisis. So let's all have our immune systems working well. There seem to be some specific pathogens linked to Alzheimer's. So as I was talking about, the herpes simplex virus has been detected in the brains of elderly individuals with Alzheimer's. Then the gingivitis also found in the brain tissues of Alzheimer's patients. So seems to be associated with increased amyloid beta accumulation. The proposed mechanism is that it's systemic inflammation plus maybe some direct brain invasion. Again, going back to this whole leaky blood-brain barrier. Lyme disease, so Borrelia infection, if untreated, leads to chronic neuroinflammation. It can lead to some issues. There have been cases of that being reported. And the idea is that maybe you have increased dementia risk due to persistent immune activation and glial stress, and of course post-infection syndromes like long COVID. Polypharmacy and sensory health. I mean, polypharmacy is probably the easiest thing to address, can mimic and worsen cognitive symptoms, but it seems like right now we are much more aware of it. We're always obviously checking for drug-drug interactions. Your pharmacist should be flagging drug-drug interactions. They have software for it. So that should be good. Sensory health. I mean, that seems to be a no-brainer, no pun intended, and seems to be working very well. Like vision and hearing loss, they seem to increase dementia risk due to just reduced cortical stimulation, cognitive load. But hearing aids and vision correction can help. One of the things that seems to be helping. A lot of just really interesting studies looking at that and how that seems to be one of the preventative strategies that can work. So the idea is that sensory interventions can reduce metabolic stress on brain regions. required to compensate for it. Physical activity. I mean, this is probably one of my favorite, dancing as an intervention tool. I mean, dancing is actually a very complex thing to do. You have to coordinate a lot of things. It's music, it's hearing the rhythm, it's hearing things, it's movements. It's a very complex activity, and it seems to be actually associated with improvement of physical, emotional, and social health in people with dementia. And that's just lovely. Another very interesting one is intranasal insulin therapy. And the rationale here is that although neurons obviously use insulin-independent glucose transporters because they do not want to be limited by things like that, astrocytes and like metabolic regulation support staff are insulin sensitive. So you can give a boost to this. And what I've seen in the literature is intranasal delivery. So just basically squirt it up your nose, and it seems to bypass the blood-brain barrier this way and deliver insulin directly to the central nervous system. And there was this even, there's some clinical evidence. There was a randomized controlled trial. They showed that like 4 months of insulin actually improved delayed memory and functional abilities. Although these are from a few years ago, and I've not seen much literature follow up from that, but I'm hoping that this is something that is still going on. Like in this paper, they were showing that in older participants, it compared to placebo, intranasal insulin actually increased perfusions through the occipital gray matter. And that is so something that maybe can give a temporary boost. But again, we're looking at now, it seems like a lot of separate pieces that need to be coming together in terms of really mainly prevention. This was interesting, photobiomodulation therapy. It keeps coming up whenever I look into anything dealing with metabolism. But the idea here is that you use near-infrared light to stimulate mitochondrial function by targeting particularly cytochrome C oxidase in the electron transport chain. And so maybe this is something that could be addressing this initiating event of mitochondrial dysfunction, or at least one of the key ones, the upstream ones. Seems to boost ATP production and reduce oxidative stress in neurons. There was this relatively recent paper from 2024, and they were looking at this and preliminary clinical studies that they show some cognitive performance improvement here. I mean, it was only 12 weeks, so longer ones would be obviously needed, but this was promising. One of the current limitations right now, it seems to be hard to really fine-tune it, like how much to give and how often and etc. So treatment parameter optimization is still in progress, but could be something interesting. And then probably the most interesting one that I've seen is klotho. And klotho is a hormone-like protein, so it's produced in the kidneys and the brain, and generally it circulates through the body in a soluble form. It's very nicely named after Clotho, Greek fate, who spins the thread of life. And it showed some really interesting things, like mice overexpressing Clotho live 30% longer. But if you remove it, it causes premature aging. So this is a wonderful cause-effect experiment, like showing actually if you add it, it works. If you remove it, it causes issues. We do know that Clotho levels decline with age, just natural Clotho levels decline with age, seems to be correlating with increased risk of cognitive decline. So quite a few papers on that. Super exciting is that exogenous Clotho administration seems to be improving cognition in mice and in non-human primates. So not just mice. Like, for example, in rhesus macaques, single ejection led to improved memory for 2-3 weeks, even though it doesn't cross the blood-brain barrier. This was just such an interesting and exciting paper. The proposed mechanism that I've seen, so maybe it stimulates the release of platelet factor PF4, so it has a lot of properties. I know it from my work on angiogenesis and as an antiangiogenic factor, but clearly it has multiple factors. But it seems to work also in knockout mice, so it's clearly not the only mechanism. So maybe necessary, not sufficient. Well, actually not necessary, but maybe sufficient. Don't know. And the idea with Clotho is maybe it's like a resilience enhancer. So it's not a disease-specific treatment, but it maybe helps neurons withstand the challenges of metabolic issues better. I saw an analogy, like it maybe acts like a little helmet that shields neurons from insults. Preclinical data are very encouraging, but obviously a lot of clinical work is still needed. Okay, basically we're almost there. So to summarize some emerging therapeutic approaches that I've been looking at. So intranasal insulin seems to be that mechanism of action is really astrocyte support, but seems to have at least some memory improvement in patients with MCI. Clotho seems to be absolutely very, very interesting. I'm looking forward to seeing a lot of stuff on that. The newer generation diabetes drugs like SGLT2s, GLP-1s. So it seems like at least so some retrospective analyses are very promising in terms of people who ended up developing dementia while on these drugs versus not, or not developing dementia. But ongoing trials, not entirely clear mechanisms of action. Non-steroid anti-inflammatories, that was really interesting. But if you think about that as being kind of more of a systemic anti-inflammatory, maybe that's the string that it plucks in that big diagram. And of course, as I mentioned, photobiomodulation, so maybe actually targets increased so energy metabolism. Very cool. Finally, I just want to talk a little bit about early detection of dementia via biomarkers. And so why does it matter? Well, it's often diagnosed after already significant damage has occurred. But there are a lot of things that actually precede it, and a trained neurologist can really note like subtle motor changes that can precede memory symptoms by many years even. So there is a window for early intervention if you actually find this. And some of the biomarkers that these specialists look at, like gait speed, gait variability, so like inconsistent step timing, issues with dual task performance, postural instability. Some of these things are very minor, but again, a trained specialist can usually detect them, and that could be an important early sign. Well, and now, of course, in the age of the machines, there is some technology-enabled monitoring that can happen. Like there could be wearable sensors that can track step cadence and postural sway and gait asymmetry and freezing, things that would also have to be specific to an individual as you're tracking how their particular markers are changing. Maybe there's going to be some, it seems like there already work on AI-powered gait analysis, which would use machine learning to like detect early deviations from normal patterns and maybe predict some dementia risk with like more accuracy. Another one that I was seeing is retinal imaging, because retina is obviously extension of the brain, and retinal changes can seem to mirror early neurodegenerative vascular damage seen in dementia. If you think of it in terms of vascular issues, obviously the retina are highly vascularized. It could be a good marker for the emerging vascular issues. And there are some very interesting imaging tools that I'm not even going to try to pronounce here, but they are looking at different aspects like thinning of nerve fiber layers or changes in blood vessel diameter and density, enlarged avascular zones. So somebody with Alzheimer's versus cognitively healthy. Can this be actually detected earlier? And of course, we have AI algorithms, so hopefully they can maybe help us or help professionals detect earlier and analyze subtle retinal features, maybe predict some symptoms so you can start with some interventions. Because it does seem like, once this damage is already done, it does not seem, from what I can tell, to be reversible. Maybe it can be delayed, but it does seem like the best thing that can be done is really trying to prevent this as much as possible. And the earlier we can find it out, then the better. And then we can start on all the things like diet and normalizing glucose and going dancing and fixing hearing and wearing glasses and I don't know, squirting insulin out of our noses. And hopefully maybe Clotho will come in, and that will be very exciting. But with all of this, maybe we can, if not prevent, then at least delay its onset, and that would be a success. So thank you for your attention on my tirade. I wrote all of this up because I needed to get this out of my head. This was actually for a different project, but I now needed to get it all out of my head. I'm not a medical doctor. I am a PhD scientist. I do mostly work on cancer and mathematical modeling. I'm not a neuroscientist, but I do go down rabbit holes, and this was really, really interesting. Just disclaimer, personal views do not necessarily represent the views of my employer or any other organization. I am not sponsored by big SGLT2 or big Clotho or anything like that. No financial support. I just really was very curious and tried to get this down and try to organize my thoughts on this. And that will be all. Thank you.